Yeah, so as Flora said, I'm going to talk about what you do once you've got your data. And it often turns out this is as big of a project, analyzing the data, as collecting it, sometimes bigger, because you're collecting so much data. And what I can say today is only just scratching the surface of a few things that you can do with this data. Uh, all right. So the first question is why do you want to record from lots of neurons simultaneously? It's worth taking a step back to think about that. Why do you want to record from lots of neurons simultaneously? Well, the first thing is, and this is really a good reason, is that it's quicker than recording them all one at a time, right? You could get, now thousands of or tens of thousands of neurons with the neuropixels probes. Doing that one at a time would be completely impossible. So even if you analyze your neurons one at a time, it's still way worth having these sort of electrodes. But of course, also there are questions you can't answer recording your neurons one at a time, which are mainly to do with the correlations and relationships between the neurons. But these are both valid reasons. So let's talk about the first one first. How do you analyze your cells one at a time? And this is still the first thing you should do, really. It's always good to start with the simple analyses, try and understand what individual cells are doing, because if you can't do that, then there's almost no chance that you can do things at a large scale. Okay, so this is still the first thing you should do with any population recording is analyze your cells one at a time. And the main thing this does is it reveals which cells correlate with which parameters of your task or stimulus. And what you normally want to do is first make some sort of a graphical plot, like a raster. We'll show this in a moment, and then summarize each cell with a number, and then do statistical tests on the numbers, because you've got more cells than you can look at. You want to look at some examples, then get numbers that summarize those examples, and then do statistical tests. You've got to be careful with the statistics for reasons I'll come to. All right. So your best friend when looking at this sort of data is the sorted raster plot. If you haven't looked at sorted raster plots, then you're probably missing something in your data. You have to do this. So that and the different ways to sort the raster by latency or by trial type. We'll see what this means in a moment. So here's some data from one of the early NeuroPixels recordings made by Nick Steinmetz, when mice were doing a behavioral task with a NeuroPixels electrodes implanted. And the task was the mice were sitting in front of some video screens. A video stimulus came on. Then they had to turn a wheel. And if they turned it in the correct direction, they would get a reward. And then they would lick the reward. And what we're looking at here is an example neuron from V1 raster plot. So the x-axis is time in seconds. The y-axis, I'm assuming you can see my mouse. Let me see if I can switch the pointer. Yeah. The y-axis is the trial number, and they're sorted in a particular way. So first, let me tell you what these colored dots are. The blue dot represents the time that the stimulus came on the screen. The red dot represents the time that the mouse made a movement. So in this plot, the raster is aligned to the time that the stimulus came on the screen, but the rows are sorted by the time of the movement, right? So that's why this red points make this sort of a curve, is because they're sorted from the time of the movement. So the very earliest movement is here. This is so early, it's probably not a reaction to the stimulus. The mouse was just going to make it anyway. But then by the time we get here, we get to the main bulk of the responses. Graph gets steeper. And then here we've got the late responses. So that's the blue and the red. The yellow points, which we see on later rasters, are the time that the feedback was given. So that's either a water valve opening for a successful trial, or a white noise pulse for an unsuccessful trial. And then the purple ones are times of licks. The first time the mouse licked after the reward was delivered. All right, so let's go back to this one that's aligned to the stimulus onset and sorted by the time of the movement. And if you look at this, you can see that here we've got a band of ticks. Every tick corresponds to a spike of this one example neuron at this time relative to the stimulus on this trial. There's all of these ticks here. And if you sum them to make a peristimulus time histogram, which is the probability of a spike per unit time relative to this moment, you see a nice spike at this moment, and that means there probably is a stimulus response. And you can tell that it's a genuine stimulus response because it's at the same latency to the stimulus, regardless of when the movement happened. All right, now let's look at this one. This is time aligned to the movement, but it's sorted by the time of the stimulus. So it's kind of the inverse of this one, right? It's the same sorting of these trials. It's the mirror image, but now it's aligned to the time of the movement. And you can see the stimulus response is still there. This little band here is time relative to the stimulus response. And you can see that the response is around 100 milliseconds after. This is a different x-axis scale than that one. So you can see the response is still there 100 milliseconds after. But now you also see something here, that you've got this broader peak, this activity that follows the movement. And that's because this cell in V1 also correlates with the movement of the wheel. So as you know, these sorts of recordings plentifully illustrated, even the primary cortices have lots of correlates of bodily movements, and we're going to see that again and again. If you look now at the time of the actual reward delivery, not much seems to be happening. Maybe there's something going on here, but if we look at this one, it looks like it's perhaps more closely aligned to the time of the lick than it is to the reward delivery. Let's look at another example cell. This one is from the nucleus of the brachium of the inferior colliculus. So that's in the auditory system. Incidentally, you can see here in this the spontaneous activity that this is a bursting cell. It fires these little events where it fires lots of spikes at random times on random trials. Okay, but if we look at the visual stimulus, there's nothing going on in response to the visual stimulus. If we look now at the movement, we see that something happens, but actually what happens is the cell is suppressed by the movement. So there's no visual response. The movement suppresses the activity of the neuron. If we look now at the reward or the feedback time, be that a reward delivery or a white noise pulse, we see that there's a very, very rapid response to it. And that, oh, I'm sorry, I got this wrong. I forgot about this task variant. This isn't the reward delivery. This is a beep that says to the mouse that it's allowed to make a movement. They often make the movement anyway, but when the beep comes on, that's when the movement is able to deliver a reward. So this is a cue, an auditory cue that the mouse gets that says it's able to receive a reward, even though it's already turned the wheel before that. And the purple here is the time of the reward delivery. I'm sorry, I got that wrong. So this is the time of the beep. And you can see there's a very rapid response to the beep, which you would expect because this is part of the subcortical auditory system. It's got a very, very rapid response to auditory stimuli. And here is the time of the feedback, which is either the reward delivery, which is accompanied by the click of the valve, or a white noise burst in case of a failure trial. And you can see that, again, both of these give you a very rapid um, response, followed by a suppression, and then followed by a rebound. And this rebound might be uh, a auditory response, or it might actually be the fact that the mouse is now licking after the reward has been delivered. Here's another example from PO thalamus. This is a higher order somatosensory thalamus. There's no response to the visual stimulus, not much of a response to the motor, although it does seem to be increasing slowly in activity around this time. So there's nothing really time-locked to the movement, but it seems to be ramping up its activity. When the beep comes, nothing really much happens, but it's here when the reward is delivered. Now you see this crazy oscillation. And this oscillation, I think, is because the mouse is licking very rhythmically to get the water. And even across trials, it normally licks in the same phase. So when you average it out, you get this oscillation in activity that correlates with the movements of the tongue. Here's another example cell from CA1, and you see nothing to the visual stimulus. Here's the movement. There's nothing much to the movement. There's nothing much to the beep. But now when we get the reward delivery, again, you get this very strong oscillation in the activity, which is probably due to entrained with tongue movements. Here's another example from the striatum or caudate putamen. And here is the visual stimulus. So there's definitely a response to the visual stimulus onset. And you can see it again in this plot where things are time aligned to the movement, but you still see there's something in response to the visual onset. There also does seem to be a response to the movement. It's not a sharp activity, but there's an increase in rate following the movement. You can see it goes down and then back up again. And here, when we have the feedback time and the reward delivery, there's not so much. Another example from motor cortex, the visual stimulus doesn't give us much. The movement here is what gives us a lot. And actually, you see that sometimes there's activity in advance of the movement in motor cortex, which you might expect. The motor cortex is what's planning the movement. So it's not surprising, perhaps, that there's some activity prior to the movement. Interestingly, if you look at the times when the movement comes on early after the stimulus, there isn't very much of this preparatory activity. But when the movement is late, you have it for longer. So maybe what's going on is the mouse was preparing to move and it was waiting for whatever reason, an extra 50 or 100 milliseconds prior to making the movement. We don't see much in this example of the beep or the reward delivery, but you can really see the difference here between these trials at the bottom, which are the ones that have a movement, and these ones at the top for which there is no movement. You have this huge difference in motor cortex firing rate at the time that the beep goes off. So that's just a few examples of what's going on, and I'm going to try and well, I won't call it a live demo, but I'm going to try and show you a website. Yeah, here it is, viz.internationalbrainlab.org. This is the visualization site for the International Brain Lab, which have done a huge amount of these sort of recordings. And it's got an interface that lets you see rasters like that. And I'd really encourage you to have a look at this website and just look at so many of these neurons that you eventually get the idea for what they're doing, because there's really no replacement in terms of understanding what these neurons do for just looking at a huge ton of data. And the International Brain Lab have this website with this lovely interface that you can use to do that. Okay, So, next thing is that there are other ways to sort these raster plots. So the ones I showed you were sorted by the rows were sorted by the mean by the time of an event, right? And that's very good when you're looking um, at event times, but it's not the only way to do this sort of sorting. That sort of a sorting is very good for behavioral experiments where there are lots of events and you need to know which spikes are related to which events. But here's an example from auditory cortex from this paper by Lutzak et al., where instead the trials are sorted by firing rate. So on the left here is a ordinary raster where they're not sorted by firing rate, they are split by stimulus. So what we've got here is a neuron from rat auditory cortex, and it's a passive stimulus presentation. There are 5 different tone frequencies presented from 0 to 1000 milliseconds, so 1 second tone. And the trials here are sorted into 5 bands, each of 100 trials each, one band for each frequency. And within these bands, they're not sorted. And you see that there's a tone onset response where the tone comes on, the neuron fires. For most of the frequencies, there's also an offset response. The tone goes off, the neuron fires again. And for some of the frequencies, there's a sustained response. So here for 15 or 5.4 kilohertz, we have a sustained response. But this doesn't tell you everything that's going on because within these bands, they're sorted in chronological order that they occurred within the experiment. But over here, the trials are now sorted differently. They're sorted by the firing rate on that trial. In other words, just the number of spikes within this time window. It's looking now at just the sustained period, which is the last 800 milliseconds. And if you sort it by the number of spikes in that period, you see this huge variability. Some of the trials have lots and lots of activity, and some of them have none at all. And that's even for a spontaneous period. So there's no tone in this period. And yet sometimes the cell is firing like crazy, and sometimes it's not firing at all. There's a trial-to-trial fluctuation in the firing rate. If we look at 24 kilohertz, which is the tone that gave us an onset response and an offset response, but no sustained response, and we sort it by firing rate, we see this similar sort of distribution to what we got in the spontaneous activity. But if we look at now 5.4 kilohertz, which is the one with the sustained response, we see that there's a high firing rate in all of the trials, but still we have this variability. So even then, on some trials, there's no responses. And if we make a plot at the bottom here of the number of spikes we get, or the firing rate, versus the brain state, and by brain state, what I mean is the amount of oscillation that's going on in the population activity. We'll see that in a moment, what I mean by that. You see that there's a correlation. So even when there's no stimulus or a non-preferred stimulus, you have a correlation of this cell's activity with brain state. And when there is a preferred stimulus, you still have a correlation with brain state, but the slope has increased. So the number of cell spikes fired by the cell depends on an interaction of the sensory stimulus and the brain state at the time that's going on. And we got all of this by doing this sorted raster plot, where rather than being sorted by chronological order, it was sorted by the firing rate on that trial. So the message I want to convey here is that these rasters are your best friend. They're the first thing you should do. You should spend literally days just looking at rasters of your data until you feel you've got some understanding of what's going on. That data Nick collected that I showed you before, we spent several hours in an airport waiting for a plane, just going through raster after raster on his laptop. And this is how we really got some understanding of what's going on in the data. There's no replacement for just looking at the data for days on end, honestly, days. Okay, but then there will come a time that you've looked at it enough, and now you need to do a confirmatory analysis. You need to go from those subjective impressions you get from the raster plots or what have you to a statistical conclusion. You need to do a formal statistical analysis and get a p-value. And you've got to be careful with this sort of data. And there's two reasons for that. The first is that simultaneously recorded cells are not statistically independent. So if we go back here, right, this neuron has a correlation with brain state. Well, so do all the other neurons you recorded. They also have a correlation with brain state. That means they're not statistically independent because they all depend on the same thing. And that means you cannot treat them as if they were independent when you do a t-test, for example. If you did a t-test treating the neurons you record as statistically independent, you'd get an absurdly significant p-value, even if you had no genuine effect. You cannot treat all the cells as statistically dependent. You can't do a t-test where the cell is treated as an independent variable. Instead, what you have to do is use the subject or the experiment, the mouse you're recording or the experimental session as the unit of independent variability. You get your data for every experiment, and you create one number per experiment that measures the size of the effect that you're looking for, and then you use the experiments as the independent units in your t-test to ask if your experimental group is greater than your control or is greater than 0 or something. You can also use a linear mixed effects model, which is kind of a development of the ANOVA that allows you to deal with this sort of dependence. But these are a bit finicky. They've got a lot of parameters. Sometimes the answer depends on what parameters you use. So the best thing is to use a simple statistical test, but using the subject or the experiment as a unit of independent variability. The other thing, and this is an even more pernicious problem, is that different moments in time are not statistically independent. So for example, if your electrode drifts, you can have slow changes in firing rate that are nothing to do with slow changes in your experiment. And this was demonstrated beautifully by Guido Maya, who wrote a tongue-in-cheek but very important manuscript called Neurons in the Mouse Brain Correlate with Cryptocurrency Price, a Cautionary Tale. And what he did is he took data from the International Brain Lab and made a plot of a decoding of from the neurons recorded simultaneously in visual cortex, posterior medial visual cortex, anterolateral visual cortex, and hippocampal CA3, use the neural population activity in those regions to predict the instantaneous value of Bitcoin and Ethereum, and found that if you take appropriate weights, you can very accurately decode the instantaneous price of these cryptocurrencies. But as Guido calmly stated, mice almost certainly lack the capacity to read and interpret complex financial data. So this isn't a genuine thing. It's a warning. It's a warning that if you've got your neurons whose firing rates change slowly because of things like electrode drift or changes in state, and the signal you're trying to predict changes slowly, such as the price of Bitcoin, which goes up or down at moments. It can even have a bit of a crash, but so can some of the cells. You're able to predict the activity of those cells, the activity of the cryptocurrency price accurately from the firing of your neurons. This is obviously absurd. So you've got to come up with some statistical method that cannot do this. If your statistical method can predict cryptocurrency prices from neurons, it's obviously can predict anything at all. It's utterly invalid. So you need some other sort of statistical method that is valid. And the reason this is not valid is because it makes this assumption, which is that you have independence between moments in time. You don't have that independence because there are slow changes in rate, and things like the t-test assume, or the f-test for correlation, assume that you have a independence of samples. So it predicts Bitcoin and Ethereum, which have these slow drifts, nearly all the time. Nearly all of the neurons show some significant correlation with those cryptocurrency prices. But if you take an actual random independent time series, just generate random numbers for every moment in time, it can't predict those. So the problem is that the neurons drift slowly and they're not independent, and the Bitcoin price also drifts slowly. So you can't, so you can predict the slowly drifting Bitcoin prices, but not the true random numbers. If you really want to test now whether neurons are significantly correlated with a slowly changing variable, sometimes you have to do that. So for example, if you want to test whether the firing rate of a neuron correlates with the wheel velocity or the movement or the licking rate or whatever, it's up to the mouse. You can't randomize it. It's not random and it changes slowly. You need a method that is able to deal with that. And two that I'd recommend are the permutation, the session permutation test and the linear shift test. And in this analysis, you see that if you use those methods, you don't get lots of false positive results. You only get 5% false positives, as you should by Fisherian statistics. So there's details of those methods and several others in this paper, Nonsense Correlations in Neuroscience, that explains first what the problem is of these nonsense correlations and second how to get around it. Okay, so that was about recording neurons one at a time and analyzing neurons one at a time, and it's still the first thing you've got to do, but it's not the last thing you've got to do. So there are questions you can't answer by recording neurons one at a time. And the most important one is the question of conditional independence. Because I'm short of time, I'm going to skip over some of this. Consider what would happen to understand what you couldn't do with a neurons recorded one at a time. Let's imagine that you did a simultaneously recorded session, but now you pretend that you don't. So you take a population recording and you shuffle the neurons between trials. So rather than really looking at simultaneous activity, you shuffle each neuron's response in one trial with another neuron's response in another trial. So it's as if they weren't simultaneously recorded, but you glued them together from different recordings. How is this different to the real thing? So let's look at an example. So again, this is from this paper in auditory cortex. So here we've got the tone coming on for 2 seconds. Here are we've got 4 neurons response to a tone of one frequency. And you see this neuron comes on for a few hundred milliseconds. This one comes on briefly. This one's suppressed. This one has a sustained response. So what's going on at the population level? Well, if we look at the shuffled population, we see something like this. So here now is a raster. In the top raster, and all the ones we were looking at before, there was 1 row per trial. But here there's 1 row per neuron. And we're looking at what the population would look like if it's shuffled between trials. So when the tone comes on, there's a lot of neurons with these onset responses. And then in the sustained period, you got 1 or 2 with these sustained responses. That's what you'd think it would look like if the neurons were independent of each other. In fact, what it actually looks like is this. It doesn't look a bit like this one. So what we've got in the population is these bursts of activity that occur at different moments, right? The tone comes on, you've got a burst of activity at tone onset, but it doesn't really look very different to these bursts of activity happening at other times, right? It just happens to drive it reliably. And it doesn't look a bit like this. The sustained activity of this cell isn't genuinely sustained. What it really is an increase in probability that the cell will participate in these population bursts. And mathematically, you can get at this idea using conditional independence. So neurons are statistically independent of each other if their probabilities multiply, and they're conditionally independent if the probabilities of different neurons multiply conditional on everything else, like conditional on the trial, on the stimulus, etc., so that the probability of two neurons having a certain spike count is independent, but only on the same trial. They can have a common dependence on the stimulus, but conditional on the stimulus, they are independent of each other. That's called conditional independence. So if you had conditional independence, you didn't really need a simultaneous recording. So what we want to understand is, how is conditional independence violated? Because that's what tells us that we actually did need to do a population recording. All right, so the easiest way to do that is to look at spontaneous activity. Spontaneous activity is when there's no stimulus. So any correlations between the cells are surely not conditionally independent, because there's nothing for them to be conditionally independent on. So let's just look at spontaneous activity. And here we're going to be looking just at the cortex. All right, so this is a illustration of what a population might look like. You've got time on the x-axis, you've got cell number on the y-axis, and every row is a different cell, and these little tick marks represent spikes of those cells. And we've got a big correlation between these cells. What you can do is, for every moment in time, count the number of spikes fired by any cell, and that gives you a number, which is the population rate. It's the instantaneous summed activity of the local cell population. You do that at every moment in time, and you connect the dots, and you get a graph here, which is the population rate as a function of time. And there are these fluctuations. We tend to call the times of increased activity an up phase, and the times of decreased activity a down phase. But really, they're not binary. They're continuous fluctuations in activity. And the amount of fluctuation tells you about the correlation between neurons. So this is a formula from probability theory. The variance of a sum is the sum of the variances of the variables plus the sum of the covariances of every pair. This is just a mathematical fact, but it has an interpretation for neurons. So if we let xi be the instantaneous firing rate of cell I, then the variance of the sum, the left-hand side of this, is the variance of the population rate, right? So in this case, we had the population rate going up and down. It has high variance. This term, the sum of the variances of the individual neurons, is what you expect if the neurons were independent. And this term is basically the mean correlation between neurons. So if the signed mean of correlations is positive, then this variance is going to be bigger than this sum. So what that tells you is that the variance of the population rate is largely determined by the covariance. So when you've got small variance, that tells you that the correlations are weak, and we call that a desynchronized state. When you've got large variance, it tells you that the correlations have a positive mean. We call that a synchronized state. So here's some examples of what that can look like. Um, uh, Again, it's simulated data. Here's the case that all the neurons are independent. The population rate histogram follows a Poisson distribution, and the correlation histogram is centered at 0 because the neurons aren't really correlated, but by chance, you'll occasionally see a detection of positive or negative correlation. There's low variance of this, and the mean correlation is 0. Now, if we look at the case that was just illustrated, that the neurons are fluctuating up and down together, we have a much larger variance of the population rate, and the correlations have a positive mean. So we have high population rate variance and a positive mean correlation. This is the synchronized state. We can also have desynchronized states. So this is desynchronized when they're independent. This is synchronized. But we can also have desynchronized state where the neurons are not independent. So in this case, we've got different neurons firing at different phases of an oscillation. This is like what you get in the hippocampus with the theta rhythm. The summed activity is constant, or it has a low variance as it did when they were independent, but the correlations mathematically have to have a mean of 0 by that equation we just saw, but now they have a wide distribution. Some are positive, some are negative, because they fire on different phases. You can also have this organization of cell assemblies where you've got different groups of cells firing at different moments, again with this low variance, but with a wide distribution of correlations. So the brain state is desynchronized when you have low population rate variance and a 0 mean correlation, and it's synchronized, and that's this top one, but also these bottom two, and it's synchronized when you've got a high variance and a positive mean correlation. All right, and in awake subjects, you see a continuum of these states between the more desynchronized and the more synchronized states. The example I showed before, it was from a synchronized state. I'm nearly out of time, so I better hurry up. Yeah, I'm going to skip that. All right. What do you do when you've got so many channels? Now, now is when we're getting to, actual cutting edge research. What do you do with this sort of data? Well, that's your job to find out. But here's a nice illustration of now, 6 years ago, recording in the brain of a mouse. The mouse isn't doing very much. This is actually a live video of that mouse. And here we see the moving rasters of 8 probes simultaneously recorded. And here you see the firing rate at different points of those probes. And you can see that there's these fluctuations of the synchronized state that we saw before in one region are really brain-wide. And understanding that structure is a totally open question. The one thing we know, though, is that it's got a lot to do with spontaneous movements. And you can show that if you take video just of the mouse's face, compute motion energy of that video. So you take a difference between successive frames, and then you do principal component analysis. You get some numbers summarizing the mouse's face, and you can use that to predict neural activity, which is shown here in the case of some neuropixels recordings. Forget the stuff on the right, but if you look here, this is the activity of the actual neurons sorted in a raster, again in a different way, now using some software called raster map that was developed by Stringer and Pachitariu. These are sorted so that correlated neurons are close together. This is the actual data, and this is the prediction we get if we predict this data from the motion of the face. So it's not perfect, but it does a pretty good job of capturing what's going on in the data. We heard about the up and down phases, but that's not what this face motion is. Here we've got time on the x-axis and the neurons recorded on the y-axis, and it's blown up in these little bits here. At this point in time, the mouse isn't doing very much. It's drowsy or maybe even asleep. And this is where we see the up and down phases, these moments when the cells in visual cortex, at least, stop firing. And if you look at other regions, frontal cortex, even thalamus and hippocampus, not so much the midbrain, you see that these down phases of visual cortex are still present in the other regions, although they're weaker. So the visual cortex, as other sensory cortices, seems to have particularly prolonged and deep down phases. It's a question that we still don't know the answer to why. But none of this is due to the motion of the animal, right? These are genuinely spontaneous fluctuations. This is where we have motion of the face, and you see we've got these correlations with the face motion, but they're not the same as the spontaneous up and down phases. And you can see at this moment, the mouse stops moving its face, the blue face motion goes to 0, and now the up and down phases start, right? So the face motion correlates are different to the up and down phases, which is spontaneous. And if we look at correlation matrices, here we've got a matrix of correlation coefficients between neurons at different time scales. Here we've got the correlation coefficient. So red means these two neurons are correlated. It's neurons by neurons. White means they're not correlated. Some of them are very correlated with each other, but hardly any are negatively correlated when we take a time bin of 50 milliseconds. So with a 50 millisecond time bin, we're capturing these up and down phases going on and off, and all of the neurons tend to be positively correlated with this. But if we look here at a 10-second time scale, now it's different. Now we've got some positive and some negative correlations, and that's because at 10 seconds, we're looking at the correlation with the movement of the mouse, not the correlation with the up and down phases. And if we look now at the correlation coefficient between two cells as a function of time, you see this very sharp peak of width around 50 milliseconds, but the prediction from face motion is over the scale of several seconds. Okay, so the spontaneous activity, this is sort of how it looks at the moment, that movement correlates are everywhere, and this is a huge caveat for your recordings. If you see a reward signal, maybe it's not, and you think you've got some correlate of the cognitive correlate of the reward, maybe it's not the reward, maybe it's licking. If you've got a choice signal, maybe it's not really a choice, maybe it's a movement. If you have a go, no-go task, you can't tell those apart. If you have a two alternative forced choice task, maybe you can tell them apart if you've got a correlate of one side versus the other. If you've got an apparent correlate of cognitive state, maybe it's really a change in spontaneous movements. Earlier, I talked about nonsense correlations. That's when you have an erroneous correlation between two slowly moving variables that aren't actually related. That's not what I'm talking about here. It would be a genuine correlation of neural activity with cognitive state, but nevertheless, they're both caused by a third variable, the movement. So these movement correlates are everywhere, but they're not the same as up and down states. As the spontaneous fluctuations, they're slower than that. Faster spontaneous patterns also exist, such as the gamma rhythms and so many other types. How are they coordinated between brain regions? This is completely open. This is your job to find that out. And how all of this ongoing activity interacts with what the responses to sensory stimuli and the command of movements and all of those things, all of this is really basically completely open. All right, so to summarize, the methods for analyzing population recordings are in their infancy. We are really only just beginning to figure out how to do this. We've got this flood of data. We don't know what to do with it, and we need your help. Start with single cell analyses. Look at your data as much as possible. make sorted rasters, but then when you come to analyze the data, remember that your simultaneously recorded cells and your successive time points are not statistically independent. Be aware of nonsense correlations. Remember that motion correlates are everywhere, and most of all, have fun, because this data is just amazing to have. It was unimaginable that you would have data like this 20, even 10 years ago. So really enjoy it, because it is a unique position to be in. All right.